Okay guys, this is our first screencast in the blood unit. This is going to be a screencast that covers the Kastle Meyer test. The Kastle Meyer test is a presumptive test for visible stains that we find at a crime scene that we suspect might be blood. This is going to help us differentiate between things like ketchup or paint or ink or any of that sort of stuff. So again, remember what a presumptive test is. A presumptive test tells us what something likely is, or it tells us what something definitely isn't. They're usually quick, they're easy to learn, they're easy to interpret, and they don't require a lot of special things. And as we learn about this test, you'll see that this definitely satisfies that requirement. So another nice thing about the Kastle Meyer test when we start talking about blood is it's a non-destructive test. When we're looking at blood evidence, we're always going to try to get DNA from it. We're always going to try to do some blood typing from it. So this is a non-destructive test, and it's non-destructive not because of the test, but because we take a little bit of the blood sample on a cotton-tipped applicator or a Q-tip and test that. So again, we're not ruining the whole sample. We're picking up a little bit of the sample, and then that's what we're going to test right there. So don't add any of these test reagents directly to the blood stain. You're going to pick up a little bit of blood stain with the Q-tip, and that's what you're going to use. So let's get into it. So it's a pretty basic test to carry out. The methods here, so the first thing you're going to do is you're going to moisten a CTA. CTA is a cotton-tipped applicator, fancy name for a Q-tip. So you're going to moisten a Q-tip with 1 drop of distilled water. Again, you're going to need the dropper bottle that's labeled DH2O. At that point, you're going to swab the stain. You need to swab it gently. You don't want to pick up a lot, and I'll explain why you don't need to pick it up during the demonstration video or why you don't need to pick up a lot. But you need to basically get the Q-tip wet. You need to kind of lightly roll it over the stain. At that point, you're going to add a drop of ethanol here, or ETOH. That's the shorthand for ethanol. You're going to wait 2 seconds. You're then going to add 1 drop of Kastle Meyer solution. And again, you're adding this directly to the Q-tip. Wait 2 seconds, and then you're going to add 1 drop of hydrogen peroxide. And again, then wait 2 seconds, and this is where you'll see your results. Don't forget about your controls. You're going to always need to start out by running a positive and a negative. That way, again, we know what a positive test looks like, and we also know that our test is working. It's also showing us what a negative reaction looks like, and what to make sure we don't have any contamination in our reagents or our dropper bottles or our Q-tips or anything like that. A little bit of background here as to how it works. So here's a diagram here. I'll explain this. There's 2 things that aren't on here, the water and the ethanol. The purpose of the water on the Q-tip is just going to allow us to kind of moisten the stain to pick a little bit of it up. We're not going to be successful in picking up part of our blood stain if it's dry and we run a dry Q-tip over it. So a drop of water gets added to the Q-tip to help us pick up a little bit of the blood. The ethanol, or the ETOH, that's going to help us break open the red blood cells. And we have to break open the red blood cells to get the rest of the test to happen. Once the ethanol breaks open the red blood cells, it exposes this right here, the hemoglobin. Hemoglobin is the part that makes our blood, part of the things that makes our blood red. It's what holds on to the oxygen and the carbon dioxide. It's very rich in iron. So the hemoglobin itself is going to react with the hydrogen peroxide. So hydrogen peroxide is H2O2. It's water with an extra oxygen molecule attached to it, or with an extra oxygen atom, I should say, I'm sorry. So again, the hydrogen peroxide reacts with the hemoglobin. And what it does is it splits that it splits the hydrogen peroxide into water, and then we end up with this extra oxygen over here. This oxygen is what we now call a free radical. That free radical oxygen then in fact reacts with the Kastle Meyer solution that you add, and that's going to turn into a nice pink color here, and that's going to be our positive change. So this is going to look very similar to our presumptive tests for the Burtonol drug. The reason you don't want to rub your stain too hard when you're picking it up is because a positive change is going to be a kind of a robust pink, but we're also picking up a red stain. So if I really scrub that red stain with my Q-tip, I'm going to end up getting some of the red on the Q-tip, and then as that gets diluted, that might start to look a little bit pink. It's a very robust pink. It shouldn't be confused for just a little bit of blood that you picked up. But again, here's the mechanism as to how the Kastle Meyer test works. It's very simple. Make sure you have that understood as well. The next thing coming up is a demonstration video of me going through the test. Again, I want you to watch both of them. After you've watched both of them, bring me your notes, and I'll give you a quick quiz, and then you can get on to carrying out the test. Okay, this is a nice short one. Go ahead and enjoy the demonstration video now.